So we're now in 2012. You've seen this before. You've seen that before. Now let's get on to it. What I'm going to talk about today specifically are experiments, biological experiments you can actually do with a microbeam, and I'm going to concentrate on protocols that we have developed here specifically for microbeam studies. Now I'm going to be around for the next couple of days, so if you're thinking about this stuff, I'll be around to talk about it. We don't need to sort of really go through it now, as what's his name? Marcelo just said. We do have to get on with it because the guys are expecting you down below. Now, requirements. I prefer to use this thing. Requirements for a microbeam experiment: preparation, preparation, preparation. You really do have to be careful in setting up your experiments. You are not doing simple, straightforward, standard in vitro studies that you are perhaps used to. The mere handling of cells for a microbeam study exposes the cells to a stress situation. Recognize that. You have to minimize those stresses as much as you possibly can to enhance the physiological well-being of your cells. So what can you do? You can start out with cell type, single cell suspensions. That's how you start. You grow your cells, you trypsinize them into a single cell suspension. You can modify them for cell cycle stages. If you start out with a plateau phase population, you trypsinize them, essentially they're in G1. You can allow them to progress and essentially do cell cycle stages. But basically, you start out with a population of cells in suspension, which you then plate on a surface. Now the surface specifically here is a thin film of polypropylene. Why polypropylene? Because other thin films that you might use, like mylar, many of you are probably used to using mylar, fluoresce spontaneously. You don't want that. It inhibits your ability to image the cells. So that's why we stuck to polypropylene. By thin, it's 3.8 micrometers thick, this polypropylene. And because majority of cells will not attach to polypropylene, like they will attach to mylar, or they will attach to a prepared plastic surface. You know what your standard plasticware is like, and you also know what the bacteriological stuff is like. You try and grow normal human or animal cells on a bacteriological plastic dish, nothing happens. That surface has been prepared so that cells can grow on it. And you have to do the same thing here. You have to prepare your cell surface, your growing surface I should say, so that cells can grow on it. So that's why this substance here is utilized, the cell tack. Now cell tack is a proprietary compound. It's actually derived from the foot muscle of the limpet. Now limpet is that shellfish that sort of sticks to the bottom of boats and has to be scraped off and what have you. So this is pretty potent stuff. And you put a thin film, there's a whole methodology associated with this, and you're going to go through part of this. It's necessary, it's not simple, and it's a requirement. You have to understand how this stuff works. So the cells have an ability to attach to cell tack. You have to prepare that cell tack in such a way that the cells will not detach. You do it incorrectly, and you take your dishes into your microbeam, you look down the microscope, you're just about to go onto the microbeam. Get your first dish out and you're looking, oh my God, the cells are coming off. What have you done wrong? What have you done wrong could well be that you haven't put the cell tack on appropriately. So you'll be putting cell tack on dishes, and you will all be doing it appropriately. Now, you can control your cell numbers per dish. You have your cells in suspension. How many cells can you put in a drop? You can put 10 cells in a drop. Now by a drop, I mean 2 microliters. We're not talking large volumes here. We're talking small volumes. You're being careful at every step along the way. So you can put 50 cells in 2 microliters. You can put 500 cells in 2 microliters. You can put as many as 5,000 cells in 2 microliters. You put 5,000 cells in 2 microliters, the cells will attach to the cell tack and they essentially will be near confluent almost immediately because there's so many. But by doing that, you're controlling the distance that these cells are apart. So you can control the distance that cells are apart on that dish that where you are irradiating those individual cells by controlling the cell numbers in that drop of medium that you initially plate on your dish on that cell tack surface. So there are a lot of things you can do beforehand. So you've thought all about this before you do your experiment. What sort of experiment am I going to do? What do I need to do to ensure that I can get the results that I want? No, so that's the sort of thing that you can do. You can look at these are just a couple of examples. If you're interested in looking at DNA damage foci, okay, you irradiate with your microbeam, you ask yourself about the expression of 53BP1, gamma H2AX, or one of the other markers of DNA damage repair. These of course can be done in cells in situ. You can fix your cells on this surface. You've then got to take it off and put it on a microscope slide so you can examine it, but that is something that can be done. It's not simple because you are dealing with a 3.8 micrometer thick of polypropylene, thick piece of polypropylene. So this sort of waves around like a hair in the breeze, and you're trying to get it to come down smoothly on top of a glass slide so you can then look at it down your microscope. But yes, all this can be done. Now if you're interested in mutation, which is a relatively rare event, you might have to irradiate 1,000 cells, 10,000 cells to get a mutant event. So what you need here, if you irradiate cells, like just as Tom was talking about, he's talking about irradiating cells through the cytoplasm. And that's being done with about a maximum of about 2,000 cells per dish. I mean, you remember the slide he showed with the cell here, cell here, cell here, cell here. If your cells are too crowded, you can't discriminate the cytoplasmic areas. So they need to be spaced appropriately, and that's what I meant. You've got to think about what you want to do before you start. What's your endpoint? What do you need to accomplish in your cell preparation to enable you to achieve your intent? So you need to be able to get all the cells off. If you say you're going to irradiate 100% of the cells, you want to make sure you get off 100% of the cells that you put down. Not quite as simple as it sounds. So you start out by plating individual cells. Ideally you want them evenly spaced. If you don't trypsinize them up and down so it truly is a single cell suspension, you're practically wasting your time. A clump of cells on a microbeam dish is pointless. We're talking about particles that go 30 micrometers. So if you have a clump of cells, the particles aren't even going to get through, and you're not going to be able to discriminate them. So clumps are bad. Now, you can discriminate cells on the basis of a vital dye. We nominally use for DNA, Herx 33342 for the nucleus. Now, Herx 33342 is a DNA binding agent. It binds in the small groove of DNA. So if you use large quantities of it, it's toxic. If you use, it'll kill cells. As you come down, what it can do is slow the growth of cells through the cell cycle. So you've got to be careful. The other thing you have to be careful about, when you are using the concentrations of Herx 33342, which will enable you to see your nuclei. If you can see your nuclei, all you got to do is make sure you miss the nuclei. And if your nuclei are in this direction, you go here, here, you can hit the cytoplasm. You don't really have to use a cytoplasmic dye. Just have to make sure you can see your nucleus in order to do a cytoplasmic irradiation. But the Herx 33342, when you put it on, You put it on and then you do your cells within a few hours. Because if you leave it on and your cells continue to grow in the dish, they'll kick it off. As they replicate, as the DNA replicates, it actually pushes the molecule out of the groove and off. So you have a time restriction. So time post-plating, you have to allow your cells to drop, to spread, and if need be, monitor the progression through the cell cycle. What sort of things can we look at? Okay, here's the easiest one. You've plated cells. You know how many cells you plated, and if you think you've plated 200 cells, in your drop, you can look at it. Just look down the microscope, and you ask yourself, how many cells are there? Okay, you can do this 2 hours later, 12 hours later, 24 hours later, whenever. So you can fairly easily monitor cell growth just by using your eyes and looking down a microscope. There usually are not enough cells in a microbeam dish to justify taking the cells off and using something like a culture counter. You can use a hemocytometer, but culture counters no. This one, we mentioned that previously. You're interested in DNA damage foci, you're interested in the induction and kinetics, so you allow cells to progress. You've handled them, you put them down, irradiate them on your microbeam, you've put them back in the incubator, you pull them out of the incubator, fix them in situ, and look at them. And what you are going to see, of course, is live cell DNA damage focus induction. You will see that. It's very cool, totally cool. And here, of course, you have to have a cell which will enable you to see DNA damage induction. In this particular instance, the cell has a GFP, a fluorescent green, a green fluorescent protein tagged DNA damage repair protein, either XRCC1, which is a single strand break repair protein, or 53BP1, which of course is a double strand break repair protein. So the cell nucleus, you can see the cell nuclei just looking down the microscope, image them without any dye. In this instance, you don't need your dyes because your cells are already tagged. And look at the redistribution of this protein, which you will see at sites of DNA damage. The next, another simple one is micronuclei. What are micronuclei? They're pieces of DNA which come about because chromosomes have been broken, rearranged, and you finish up with an acentric fragment. A piece of chromosome without a centromere is lost from the cell at division. Now, micronuclei, you can't look at that in the cell that has been irradiated as such. You look at that at the next division. So micronuclei have to be looked at when the cell has advanced through the cell cycle into the next division. So the micronucleus assay can be done in situ, or you can take the cells off, put them in culture and look at them in time. If you put bromodioxyuridine on the cells, you can, the BRDU of course is taken up into the DNA during S phase. You can fix them and look at these two endpoints essentially simultaneously at the same time. You can monitor cells that are undergoing apoptosis either by morphology or by the use of antibodies against one or other of the proteins that are involved in apoptosis. Another one that we have done is the cells can be removed and then you do the COMET assay, which you're probably aware of. Bromodioxyuridine, immunofluorescence, cell cycle progression. We can look at cell site-specific protein expression in situ, or if you've got enough cells, you can actually remove the cells and put them through some sort of monitoring system. We've also done single-cell gene expression via RT-PCR, which requires you to take cells off one at a time to put them in a microcentrifuge tube one at a time and then do an RT-PCR reaction. Okay, examples of experiments that were done here at Nevis fairly early on actually. This is, oh good, I can't remember when, a long time ago. 3 different cell lines. The cell line that Tom talked about, the A sub L cell line, which is a human hamster hybrid cell line. HeLa cells, which the cells that Henrietta Lacks provided to the scientific research community, about a book of which has just been written about Henrietta Lacks and her cells. And C3H10D1/2 cells. These are hypertetraploid mouse fibroblasts that are used for the in vitro transformation assay. So these cells can be used for different endpoints, mutation in this instance, transformation in this instance, and these just because they're the standard weed that most people who are indulging in cellular biology use. They are. You've got to be careful with HeLas. They'll take over other cell types if you're not careful. So okay, we're going to use a microbeam to hit the cell nucleus. And if we want to hit the cell nucleus with exactly one particle, we can. You just heard Gerhard tell you so. That your physicist, your friendly physicist, you can go to him or her and say, I have these cells, I want to hit the nuclei with one particle. Sure. He will agree and he will set it up so that you can. You will look down the microscope and you'll see the image of nuclei. One of the first things that you will notice, as I'm sure you know, any of you who have looked at cells in culture, these are not uniform in size. Of course they're not uniform in size. They're at different stages of the cell cycle. Many things are going on. So there is no such thing as a mononuclear cross-sectional area. This is a mean. You measure them. You measure the nuclear cross-sectional area in a population. So this is the mean for these 3 cell types. Now, what I'm going to do is give these cells one particle each. Okay, right now you say, well, one particle, what is a particle? Well, you know what a particle is. It's 1 alpha particle. What is the dose? Good question. Because what you might want to do is compare your microbeam known numbers of particles with random particles given as a function of dose. That's a comparison. That's a straightforward comparison that you might want to do. And the dose per particle is here, here, here. Now the first thing you see is, oh, they're not the same. Why are they not the same? Because the way you determine the dose is via this equation here, which I must admit I had intended to put in the form of a slide but forgot. So I actually used a piece of chalk. You've probably all forgotten what this stuff is, but this is chalk. Maybe when you were in kindergarten or primary school, your teachers actually wrote on the board, a blackboard or even a green board with chalk. And this tells you the relationship between dose, LET, and particle number.
John, the 6.24 is a constant, right?
That's the constant, yeah.
From?
Andrew.
It's essentially converting current numbers.
Yeah. No, it didn't come from Gerhard. It's, I can't remember who. I think this one, in point of fact, this one came to me first from Stan Curtis.
I use that, yeah. All the NASA folks, we use that for this.
Yeah.
They do the
Yeah.
conversion.
Okay, well, just sort of let's go through this very quickly. So if this is the relationship, dose in grey over LET in kV per micron by this constant, 6.24, equals fluents per micrometer squared. What do we want to do? We want to give a fluence of one particle. Let us assume that our micrometer squared, our mean nuclear cross-sectional area, is 100. Okay? So this is 1. This is 100 micrometers squared, which is pretty close to 2 of those cell lines there. Our LET, the LET here was 90, but let's keep it simple. It has to be simple. I am a biologist, remember. I'm not a physicist. I leave the sort of using complicated numbers up to Andrew and people like him. Let's say the LET is 100 keV per micrometer. So what do you have? You can cross these out. And what you finish up with is 1 over 6.24. Now this is a made up number, but this is how you get to this. That's all I'm doing. I'm just giving you that as an illustration.
So as a suggestion, it would be great if you can use this formula for your proposal. Thank you very much.
Okay. So what do we do? That's just how you do this sort of thing. What did we do? We actually looked down the microscope and looked at micronuclei and or chromosomal bridges. A chromosomal bridge is where 2 chromosomes have broken and rejoined with each other. Then they've gone to opposite poles. You have a dicentric chromosome, dicentric, 2 centromeres, 1 centromere goes to one pole, 1 centromere goes to the other pole, and they bridge. They extend across from those 2 daughter cells, those 2 progeny cells. So that's where you get the bridge. The bridge is the direct consequence of radiation exposure and reunion between 2 different chromosomal entities. And obviously, if you finish up with something that's got 2 centromeres, which shouldn't have 2 centromeres, something else has none. And the bit that has none is this. That's your micronucleus or acentric fragment. The acentric fragment of chromosome is left out of that nucleus that is reconstituted after a normal mitosis. Very simple process, straight out of basic cell biology. So this is the incidence per particle, and then if you convert it to per gray, that's it there. Then if you now shift to another cell type, and remember you start out irradiating one cell. And what you're interested in is the progeny. That one cell divides into 2, so micronuclei per cell pair. This is the control level, and obviously as the number of particles have gone through the nucleus, and what they do is, and this will be true of any cell type that you put in front of the microbeam. You say you want to mark the nucleus, your image analysis system will run around the edge and it will give you the nucleocentroid. And that is the aiming point for the beam. So when you actually set your experiments up, you say you want to give a particle to the nucleus, the first thing it will do is aim at the nucleocentroid. Okay, and you can use bromodioxyuridine and okay. So we're now going to shift just momentarily to DNA damage. This has been mentioned previously. Some of these things you've seen before. 53BP1 here, gamma H2AX. These are just a couple of molecules that are actually attach to DNA. So for our purposes, they're very useful. So for gamma H2AX, what do we see? As a function of time, 4, 2 hours, 6 hours, 24 hours, you can, in other words, on a cell-by-cell basis, you can look at the induction and fairly rapid decline of these DNA damage repair foci from specific sites. And of course, we're not talking about one particle here. This is multiple. Okay? Here, 4 minutes. They come up fast. Then gradually fall off. This is one of Alan Bigelow's experiments with the microbeam. in cells that are expressing a green fluorescent protein. So the first thing he did was irradiate here. Then he waited 2 minutes and irradiated here. And this is live. This is live. So at the end of 4 minutes, this is what he saw. And what do you see? You see that essentially all this fluorescent protein that is in this cell nucleus has aggregated into these 2 sites of irradiation. It's freaky. It is truly freaky that all this protein that has been distributed around in this nucleus has aggregated to these irradiation sites. You look at it, you wait in time, it declines. This is 15 minutes. It's all gone. It's all gone. So this whole process has come up. You have this site-specific conglomeration of DNA repair proteins. This is XRCC1, incidentally, it's not 53BP1. And it's gone. And this is what Alan has been doing for the last couple of years with these cells that express fluorescent proteins for DNA damage proteins. Okay. Now we're going to do a bit on hit and bystander response as opposed to specific responses. What is a bystander response? Non-hit cells display similar qualitative but not quantitative responses. We've just heard from Tom and from David before him about the origin of the signals. So if we're using a microbeam and you pass a microbeam through the center of a cell, and you say you're aiming at the nucleus. Yes, you are. You are aiming at the nucleus. What's going on? You hit the cell membrane below the nucleus. You hit the nuclear membrane. You hit throughout the depth of the nucleus, which in our purposes is probably 4 to 6 micrometers, up to 12 micrometers thick. And it goes through the cytoplasm on the underside of the nucleus, through the cytoplasm on the bottom side of the on the top side of the nucleus, and through the top nuclear membrane, and it goes through the medium. If you've got a microbeam, you can irradiate the medium. You can go by a cell, miss it deliberately. Now you might think, God, who'd want to waste their time doing that? If you want to really know what's going on, that's the sort of thing you should think about. I've done it, and I know it was almost a waste of time, but anyway, it comforted me that if I irradiated adjacent to the cell, nothing happened. The next thing you can do is irradiate through the cytoplasm, like Tom said. Then you irradiate through the nucleus. You can irradiate through different parts of the nucleus or membranes, trying to miss things deliberately and determine where your signals come from. So these are things that you can do. You can refine it down to pick sites within the cell which will enable you to both ask and answer the sort of questions that you might want to ask. Broad beams go everywhere. Everywhere. Whereas microbeams are precise radiation tools, you have a spatial and a temporal resolution. Nobody's mentioned temporal so far. But you, yet again, you ask your friendly physicist, I have this population of cells. I want to irradiate them through the nucleus. Then I want to come back half an hour later and irradiate them again. Same nuclei, same place. You want to move that site by 4 micrometers? Say, sure, we can do that for you. Very cool. Just think about it. Very cool. And the big thing is about the microbeam is the microbeam provided validation for the bystander response. Once this stuff came out with microbeams, all the questions that had arisen initially just went out the window. I should say, and this is not only true of me, it's true of many of my colleagues, that when the first bystander responses were reported, I said, oh, come on, give us a break. No, this is not possible. We'd been looking at charged particles, effects of charged particles for many, many years. Remember, I started here 37 years ago. And we just found it difficult to believe there was such a thing like a bystander response. We're believers now. And we were the ones who started out questioning the whole basis of it. And if it's good enough to convince us, okay, we'll accept. Okay, now I'm going to go in a little bit of detail into some specific protocols. They're in your book, so that I'm not going to read these out, but as I say, I'll be around the next couple of days, so if you have questions about these as we sort of move through them, we can go through it later. So this particular one, okay, as I said, you decide on your cell type. In this instance, they're human fibroblasts in plateau phase, so they're about 95% in G1. Trypsinism, single cell suspension, plate varying numbers in a microbeam dish, and that sort of can vary the number of cells that are in contact, enables you to ask a question, as was asked previously, about what is the bystander response? Is it a signal that passes through the medium, or a signal that passes from cell to cell, where the cells are touching via gap junctions, etc., etc., etc. And as Tom High said, it's both. No question, it's both. You want to be able to be sure that you can discriminate your cells. A nice, easy way to do it is to start out, David Brenner showed this morning, by discriminating your cells using two different dyes. So you have, it's the same population, but you split them in half, you take samples, one half are placed in this, Herx 33342, the other half are placed in this, cyto orange, which obviously is cytoplasm, orange fluorescing, Herx 33342, blue fluorescing. They are readily distinguishable based on that, the activation energy of the wavelengths that you hit them with. No question. If you use the appropriate wavelength, you'll see these and not those. If you use the appropriate wavelength, you'll see those and not those. So you can decide, I am only going to irradiate those. And you can plate these at known ratios, from 100% of one or the other to 50-50, etc., etc., etc. So that you then do this, and then you put a known number of particles through them, and you look at effects. You can, as I say, here, put them in, like so. You irradiate just one, and then, because these are blue and these are yellow, if you start out with a micromanipulator right here, you can actually remove these cells one at a time. Just let your mind wander around that, and you think, oh my God, one at a time. I could spend days doing this. Yes, you can. And then you do an RT-PCR reaction, or else you just leave them there and fix them in situ. And of course, this is quite completely time dependent. You can decide on your times. Or you can irradiate a fraction of cells. And in this way, when you do the past one, the one with the two dyes versus this one, you can get a direct comparison. Irradiating known numbers of cells versus irradiating a fraction of cells. And it's nice to be able to say that, yes, I do get the same results using both approaches. And I did that, and it's true, you do. So using the micronuclei in hit cells, micronuclei in non-hit cells, micronuclei and bystanders, David showed them some of this morning, higher than controls, it's not dose dependent. And an interesting one was this induction of the cell cycle protein, cell cycle control protein, controls the G1 to S transition. in 80% of bystander cells. These are the cyto-orange, these are the Herxt, micronuclei in the Herxt, micronuclei in the cyto-orange. So in the hit cells or the non-hit cells. You can look at a number of proteins in the same sample if you're careful. So this one, we're actually using 3 dyes and an antibody against P21, CDKN1A, CYP1, it's got a number of titles, and P53. This is the specific serine 15, P53 actually. And this mess here is a multi-micronucleate apoptotic cell. So the first thing you notice looking at this multi-micronucleate apoptotic cell is either that, yes, it is a mess, and it's in a hit cell. This cell was hit. These are the non-hit cells. And the red is actually P53. So you get essentially getting massive expression of P53 in these micronuclei. Here, that's P21. So you really can look at multiple endpoints just by flipping through different wavelengths with different endpoints. Okay, next protocol. We mentioned the single cell RT-PCR. All you need is an adjacent offline inverted fluorescent microscope. So when you're thinking about your microbeams, you want to set up a microbeam. Think about it in terms of what you might want to do and make it as easy for yourself as possible. Have your microscope, an offline microscope, right alongside. You'll see that down, you've already seen it. You've been through all the labs, you've been through all the various microbeam facilities. That's what we've got. And in talking about developing this thing with Gerhard right from the word go, essentially from where Gerhard showed that window, that's where the biologist comes in. But the biologist is basically your observationalist. So you need somebody who can see things to help your physicist along in setting up your facility adjacent to your microbeam so you can optimize the number of endpoints that you might be interested in looking at. Okay, what do you do here? You put a thin film of trypsin on these cells. You've just irradiated them. But the cells have got, they fluoresce with 2 different colors. And you want to take them off separately. Now if you put trypsin on and just leave it there, you finish up with things swimming around all over the place. Doesn't work. So you put a light trypsin. Then you come on and essentially pluck them off one at a time. As soon as they start to float and mixing in with each other, you stop. Now you can spend a lot of time doing this, and Brian Panaya, who did this, it took him 9 months to get to an 80% success rate. That's getting the cells off as individual cells and running them one at a time, doing RT-PCR for one or other of these RNA species. So you do RT-PCR, you're looking at RNA. There's another step down the way. So that explains how it's done. Results, individual cells. Each one of these is an individual cell. Use a control, of course, beta-actin, and this is your P21. You can quantify each of these signals, and each one is taken relative to that one, of course. If you do that, control, control, non-hit, non-hit, hit, hit, non-hit, non-hit, hit, hit. Are they the same? No. Is there a difference between the control cells and the hit cells? Yes. Is it uniform? Almost. It ranges from 6-fold and 9-fold increase above the control. But in the non-hit, known non-hit bystander cells, Is it the same as the control? No, not in one instance. So here you have a direct demonstration at the single cell level of a molecular response in a non-hit bystander cell. Uniform? Yeah, no, hardly. It's varying easily by a factor of 2. And that is true when you start looking at bystander responses in individual cells. Twofold variations are common. 30 minutes after, an hour after, 2 hours after, 3 hours after. This is for hit cells. So even in the hit cells, you're not getting a uniform response. You do a population response, which many people do, most biochemists, cell biologists for as many years as I can remember. Look at populations of cells. You want to do a Western blot, you start out with 10 to the 7th cells minimum to start looking at proteins. You're looking at the response of a population, but what's going on underneath that? What's important really is what's going on at the single cell level. And that's one thing a microbeam will do. It enables you to look at responses at the single cell level. So if in a tissue, for example, you have 10% of the cells are responding, that's producing whatever protein, at tenfold higher than 90% of the cells, you look at the population and you assume that something wondrous is going on. It's not. It's going on in a minority of the cells. So we need to be very conscious, I think, of responses in single cells. Irradiated cells, bystander cells. Now you saw the controls earlier. There's no question they overlap. There's no question they're variable at the single cell level. So keep that in mind when you're designing your experiments. We've done RNA, so you've decided, okay, I've had enough of this RNA business. I'm going to look at proteins. Can you look at proteins? Yes, of course you can. Can you look at them using Westerns? No, of course you can't. There's no way you'll get enough cells out of an array unless you want to spend a couple of weeks irradiating cells with your microbeam. But you can certainly use any assay that involves the use of antibodies against known proteins that are present in the cell. So what do? You can fix cells in situ at various times, post-irradiation, permeabilize them, probe them with antibodies, and then quantify that response. And this is just an example of some of the proteins that have been examined here. So you just need a couple of microscopes. You saw the microscopes. We've actually got 4. Oh, we've actually got 6 that are capable of giving us quantitative assessments of fluorochrome intensities on a cell-specific level. So we've got our offline microscope, so you need your appropriate filters. Think about this beforehand. When you're thinking about your experiment, don't recognize a couple of days after, oh dear, I don't have that particular filter combination which will enable me to discriminate between this protein with this fluorochrome and that protein with that fluorochrome. And think about your microscopes. You need to not only quantify, but you need to think in terms of rapid changes between filters and things like that. A lot of things to take into consideration, but it can be done. And a lot of it's going on right now. An example here, this is in situ, cells fixed in situ, normal human fibroblasts. This is the cyto-orange stained cell. These cells were originally DARPy stained cells. This is CDKN18, CYP1, P21. And what do you see? You see these cells, and only these cells, were microbeam irradiated through the nuclear centroid. Where's the nuclear centroid? In this one, it's probably about there, there, etc. So you're certainly not getting the expression of this protein at the site of irradiation, and neither would you expect to. What you do see is the protein coming up here, here, and here. These regions, here, 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 nucleolus organizers. You don't get a response in the nucleolus organizers. No surprise, you wouldn't expect to. And this is the non-hit bystander cell. Now, if I told you that there is less of a response there than there is any of the hit cells, you might say, ooh, well, maybe there is. In none of these non-hit cells, and remember, more than 80% of the non-hit cells are giving you a P21 signal. And this is a hit cell here. I'm switching colors now, quite deliberately. This is a hit cell, so this one was hit. This was not hit. This was not hit. And that one was not hit. And this, to me, is something that I like to see. I like to see a control. Well, no, I'm sorry, that's not true. It's not a control. I like to see a non-hit cell with no response. That encourages me to believe that the response I'm seeing here and here is real. You can quantify the response for this protein, which you just saw, for where cells were, different fractions were irradiated, or the dual staining technique, where only known cells were irradiated. And as you can see, the simplest one is this one. This is the control here, fluorescence per nucleus, measured. You're quantifying all those yellow dots. You can count those yellow dots if you want to, or you can determine the amount of fluorescence per nucleus to get the endpoint that you might be interested in. So this is a control, very little response in the controls, and irradiated 5 alpha particles per nucleus in the hit cells. Bystanders to hit are actually here. Irradiated with 10 here, bystanders to 10 there. There's an overlap. But this is a pretty dramatic difference from here to here, and there is no question that both these bystander populations are not sitting on top of that. In this instance, things are a little different. 100% irradiated there, 50% irradiated here, and 1 and 5. And as you can sort of see, you're sort of sitting on top of the control line, and then they're splitting off. But certainly, and this is the simplest one, you'd expect 10% of cells to show a response and 90% not to show a response. In other words, in this one, you'd expect it to follow this black line to there and then deviate if it's only 10% were responding. Okay, conclusions. In my hands at least, the CDKN1A gene products are highly expressed in microbeam irradiated hit cells and in bystander cells down to 1% cells irradiated. And I term this a sentinel monitor of bystander response. Something to think about. It's nice to have something which is going to express itself most of the time. So you can sort of use that as an internal control. Single cell RT-PCR show a lot of variability in both hit and bystander cells. And this nowhere near as apparent in bulk assays. So this led me to this conclusion. Cellular response may not need tight control and tolerance for variation is high. But ultimately, this is the point. Precision microbeams and the interrogation of individual cells certainly reveal a complexity of cell population responses which you might never have anticipated until you actually get in there and do it. And that's it.